proteins are very important biomolecules because most of our body is made up of proteins, and proteins are actually made by a combination of amino acids. So in this video we are going to study about proteins. Let's start. Proteins are another biomolecules, and actually these are used for bodybuilding. But we know that all the organs of body are made up of proteins only. In fact, our red blood cells are also made up of protein. Enzymes, hormones, they all are proteins. So proteins are very important to maintain and keep us healthy. Now proteins are polymers of alpha amino acids, and proteins are polymers of alpha amino acids, and they are joined together through a special type of linkage called peptide linkage. Please keep this in mind. In case of carbohydrate, we studied about glycosidic linkage. In proteins, it is peptide linkage or peptide bond, and this peptide linkage is formed between the carboxylic acid group and amine group of two amino acids. This is how they are formed. This is one amino acid, glycine. This is another amino acids. They react together. OH of this and H of this come out in the form of water molecule, and we get a bond which is known as peptide bond. This was actually glycine, and this was alanine. So this is a dipeptide as it is formed by two amino acids. So, and its name is glycylalanine. In short, it is written as GLYALY. So the name of peptide, polypeptide or dipeptide, depends on the name of the amino acids which come together to form that peptide. So in case of glycine and alanine come together to form this peptide, which is known as GlyAli. Next is again classification of protein, as we were just discussing. If it consists of two amino acids, it is dipeptide. If it consists of three amino acids, it's tripeptide, and it contains, if more than 10 amino acids, it is polypeptide. Next is, again, proteins are classified on the basis of shape of the proteins, and the one is fibrous protein. In fibrous protein, polypeptide chains run parallel, and they are held together by hydrogen and disulfide bonds. Example, myosin and keratin. Next is globular protein. Globular proteins are polypeptides actually, chain coiled around, giving spherical shape just like insulin. First is primary structure of protein. Actually, in primary structure of protein, amino acids are grouped together in the form of long chains, straight chain, and they result in this consists of more than one or more polypeptide chain, and each chain has amino acids linked with each other in a specific sequence. This sequence of amino acid represents the primary structure of protein. Different amino acids are linked together in specific sequence. That means amino acids they bond by each other through a peptide bond. They form long chain polymer, straight polymer, and that is the primary structure of protein. Next is secondary structure of protein. In secondary structure of protein, it is the shape in which a long polypeptide chain can exist. Two types of secondary structures are possible: alpha helix and beta plated sheet. This is alpha helix. It is in helical form. You can see actually the primary structure coils together due to hydrogen and disulfide bonding. This is helical alpha helix, and this is beta plated sheet. In alpha helix structure, what happens that the chain of alpha amino acids coil together as right-handed screw because of formation of hydrogen bonds. The sphere is held together by hydrogen bonds between nitrogen, hydrogen and CO, that is amine and carbonyl group, which are vertically adjacent to one another. Such proteins are elastic actually, and that means they can be stretched. On stretching, weak hydrogen bonds break up and peptide act like a spring. The hydrogen bonds are again reformed when the force is released, and the example is myosin, keratin, and that is. Next is beta plated sheet. Actually, beta plated sheet, in beta plated sheet, polypeptide chains are arranged side by side in form of sheets. The chains are held together by very large number of hydrogen bonds between carbon, that is carbonyl, and nitrogen, that is amine group, of the different chains. These sheets can slide over each other to form three-dimensional structure called beta plated sheet, and this is found in silk. Just see this. The first one is alpha helix, and this is the beta plated sheets in which these amino acids are present in the form of sheets and which are attracted together by hydrogen bonds. And this is the two type of secondary structure of protein. Primary structure straight chain, secondary structure slightly coiled because of presence of hydrogen bonding. Then comes tertiary structure. This tertiary structure refers to the arrangement and interrelationship of twisted chain protein into specific layer of fibers. This tertiary structure is maintained by weak interatomic forces as hydrogen bonding, hydrophobic bonds, van der Waals forces, disulfide bonds. For example, protein of tobacco mosaico virus is actually a tertiary protein. So simply we can say that if there is no attraction between the chains of amino acid, it is primary structure. When there is little bit of attraction between the different chains of amino acids, polypeptides, they get coiled to form secondary structure. This actually coiling becomes more complex when there is more attraction between these molecules, because of which they just coil up together in more complex way to form tertiary structure of protein. And in quaternary structure, different chains come together and they just form a complex type structure which is known as quaternary structure of protein. When two or more polypeptide chains unite by force other than covalent bonds, that means peptide and disulfide bonds, it refers to final three-dimensional shape of the result in the twisted bonds and bonding of the protein helix. It is the most stable structure. So primary, secondary, tertiary and quaternary. So again we can sum up in this way that when there is straight chain polypeptide, almost no interaction between the chains, it is primary structure. These primary chains of peptides coil together when there is some hydrogen bonding present between them, forming secondary structure. When there is more attraction between these chains, the coiling becomes slightly more complex, resulting in formation of tertiary structure. But when different peptide chains come together because of even more greater attraction between the different chains, they result in formation of a highly complex structure which is known as quaternary structure of protein. It is the most stable one. Next is denaturation of protein. Actually what happens that proteins lose their biological activity due to some unfolding of globular and uncoiling of the helix. We know there is helical structure in secondary protein and there is some coiling. Because of some reason, because of possibly high temperature, what happens that this coiling breaks. The coiling breaks because of breaking of hydrogen bonding, and because of which the protein loses its biological activity, and it is also known as coagulation of protein or denaturation of protein. And the example is coagulation of egg white on boiling or curding of the milk. The denaturation of protein may be result of high temperature, may be result of unfavorable pH or any other thing, any other harsh environment, because of which the hydrogen bonds get affected, they break down and the coiling becomes uncoiled, and because of which protein loses its biological activity. Now there are some chemical tests for the protein. The first is biurate test. On adding dilute copper sulfate to alkaline solution of protein, first of all we make the protein solution alkaline, and then in that we add copper sulfate. a violet color is developed, and this is due to presence of peptide linkage. So actually biuret test is used for detection of peptide linkage, and this test is indicated by violet color. Next is Milton test. In this Milton test, there is a reagent, Milton reagent, which consists of mercury which is dissolved in nitric acid, which results in formation of mercuric and mercurous nitrides. When Milton reagent is added to protein, a white precipitate is formed, which then turns brick red on heating. This test is given by proteins which yield tyrosine on hydrolysis due to presence of phenolic group. Next is ninhydrin test. Actually, ninhydrin is a reagent. This test is used for all the proteins, and it is basically used in case of chromatography. Whenever we use chromatography to separate the different amino acids, we use this test. This is ninhydrin test. And when protein is boiled with dilute solution of ninhydrin, or when ninhydrin is sprayed over the protein sample, a violet color is produced. Now there is a salt problem. Barford reagent is a solution of cupric acetate and acetic acid, copper sulfate and NaOH, alpha naphthol dissolved in ethanol or acetic acid and ethanol. C is the correct option. Next, select the correct statement. Protein upon hydrolysis gives alpha amino acids. Not possible, not exactly that all the proteins will give only alpha amino acids. Except glycine, all the other naturally occurring amino acids are optically active. Exactly, they are. Okay, so B is the correct option here. So students, we studied about proteins and also the structure of protein. Most importantly, we know what is primary, secondary and tertiary structure of protein. First of all, we know that proteins are nothing but a polymer of amino acids, and this arrangement of amino acid decides the structure of protein. In our next video, we will move to enzymes and vitamins. Stay tuned. Thank you.